Okay, let me turn off my video and share the screen. All right. Okay, let's wait for one more minute. All right, let's get started. Hello, everyone. All right, so I think we have something uncovered from last lecture. So let me give you a very brief review. So last time we in the last week we learned the first Mendelian law, the Mendel one, which is about the random segregation. As we learned, right? So in Mendel 1, actually, we are talking about the relationship between two alleles of one gene, a single gene. So that's why we say this is a single gene inheritance pattern. And so during the meiosis, these two alleles, the two members of the same gene on the same chromosome, they're going to segregate. They're going to segregate in a random fashion. All right, so this is all about the first Mendelian law. And by understanding, so by using this beautiful square here, we call this Punnett square, the 2x2 table, we can understand the genetics behind this monohybrid cross, all right? Okay, so then we learned the second Mendelian law, Mendel 2. So things get a little bit complicated here because we're talking about two genes. These two genes are located on two different chromosomes. Then what is the relationship between these two genes? So this is all about the second Mendelian law, Mendel 2. And as this law says, during the meiosis, these two genes are sorted completely independent if they are located on two different chromosomes. All right. So in this case, we can use a 4x4 Punnett square to help us to understand the genetics behind the so-called dihybrid cross. Right. So so when we talk about the dihybrid cross, so we have a new ratios here, right? So the phenotypic ratio, so becomes 9:3:3:1. So I hope everyone understand and remember these magic numbers, because we're going to use these numbers again and again in this class to help you understand the genetics behind a lot of experiments, all right? So this is what we have learned so far. And so, okay, so if these two genes, so the two genes that we are starting on, if these two genes are on two different chromosomes, if during the meiosis, these two genes are sorted completely independent to each other, so let me use pointer here, maybe pen, okay. So if these two genes are sorted independently, we would expect to see this phenotypic ratio, right? Based on the second Mendelian law, Mendel 2. All right, okay, now we can perform some experiment. For example, if we can copy Mendel's P-Plants experiment, we can generate a lot of data here. So this is the data we have. Okay, so by simply looking at the data, so we believe, yeah, this data kind of like agree with the expected ratio. So what is the expected ratio here? The expected ratio means given the model is correct, given given the non-hypothesis is true. So what is the non-hypothesis here? Here, the non-hypothesis is, okay, the two genes controlling these two phenotypes, they are located on two different chromosomes. If this is true, then during the meiosis, based on the second Mendelian law, the Mendel 2, these two genes will be sorted independently. And if this is true, so we will expect to see this ratio, phenotypic ratio. So the non-hypothesis we would like to test is the independent assortment. All right, if that is true, so this is also true. The expected ratio should be this. And we want to use this data to test this hypothesis, whether this ratio holds or not. All right, so we have learned how to calculate this. So we have learned how to use this data to help us to create a chi-square test to test this hypothesis last time. So we're gonna very briefly go over this again. All right, so first we can add up these numbers together to get the total number. So let me erase those, it's not very clear here. Give me a second, thank you. All right, so this is very important. So when I say something is important, so yeah, so please put the notes on your notebook or slides, because in the midterm, the first midterm, I'm gonna give you something like this to test you if you understand the genetics and the statistical theorem behind this hypothesis test, right? Okay, so it would be helpful if during the test, if I ask you a question like this, to let you do the calculation and test the hypothesis, it'd be helpful if you can use this table. So this table is super useful. All right, so the first thing you need to do is to add up all these numbers for these four categories, four phenotypes, to get the total. All right, so this is the total number of the progeny. All right, since this is the expected ratio for each category, we can easily calculate the expected number for each category, right? So we just, for example, for this number, how do we get this number for this category? How do we get the expected number for this category? So we simply multiply the total number, which is 557, 500, 557. We multiply this by its expected ratio, so which is 9 divided by 16. I hope this is clear. So this is how we calculate the expected value for this category, and the same for this one, and this one. So once we got the expected number for each phenotype category, we can easily calculate this, right? So we have learned this before last time. So here we simply fit our data with the model. So this is the data. O means the observed value, which is here. We fit the data with the model. So the model means the expected value. Expected value is the value expected. Yes, go ahead.
Hi, sorry, didn't mean to interrupt, but where did you get the 16 when you divided it by 9?
Oh, right. So that's right. So if you add up these numbers together, so that gives you 16, right?
Okay, perfect. Thank you.
Yeah, sure. So yeah, that's a good question. So you need to get the fraction of each category, and then you can calculate each numbers here, right? Okay. All right. So now for each category, you have to fit your data with the model, or think about the example I gave you last time. So you put the beautiful dress on yourself and look into the mirror to see if yourself is the data, if your data fit the dress, yes or no. So this is the way we do this calculation. So how do we fit the data with the model? So we just take the difference. We see how close they are, right? If the difference is very small, close to 0, that means data is very supportive of your model, the expected value. Otherwise, if they are quite different, so you're going to end up with a big number. All right. So for each category, you can calculate a quantity like this. And then you add up all these numbers together to get your chi-square test statistic. All right. So now, if you want to calculate a p-value, so everyone knows when you do a hypothesis testing, you need to get a p-value to make the final conclusion. So this is a chi-square test. You need to calculate the chi-square statistic, which is here. Oops. So which is here. But to calculate the p-value, you need another thing, which is the degree of freedom. In this case, we have 4 phenotypic categories, which means the degree of freedom equals to 4 minus 1. Wow, my slide is a little bit messy. Sorry about this. Let me erase this. Okay. So here, the degree of freedom equals to the number of categories, phenotypic categories minus 1, so which is 3. So now everything is ready. Okay. So we have calculated the chi-square statistic for this data, right? And we have the degree of freedom equals to 3. So the next thing is simple. So we go to the chi-square table. So during the midterms, I'm going to give you this table, and you don't have to look. So it's a closed book test, including the quiz and the midterm and the finals. All right. So yeah. So this reminded me of like on the first time in the first lectures, some student asked me whether we're going to use the Yuja system for exams and quiz. Yes, we do. So please use the Yuja system. Tomorrow will be your first quiz in the discussion session. So please use that platform, because I learned this from other professors, and everybody needs to use the Yuja. And to be fair to everyone, if someone did not use this system in the quiz or exams, so we have to take some points off. So yeah, I hope you all agree with this. All right. So the table, so chi-square table. So you're going to use this table. I'm going to provide this table for you for free during the test. All right. So so in this table, you have to look at this row. Why? Because this row represents the degree of freedom equals to 3. Because in our problem, the degree of freedom equals to 3, right? And you need to. So let's go back. So this is the calculated chi-square statistic. All right, so in this table, you need to find the numbers that are closest to the number you calculated from your data. So these 2 numbers are very close to that number, and which means your number is somewhere here, right, between these 2 numbers, and your p-value should be something between the 0.9 and the 0.975. Okay, so what does that mean? That means the p-value is pretty high. The p-value is larger, is greater than 0.9. And we have talked about this last time. So the p-value, you can treat p-value as the probability, how likely your null hypothesis is true given the data observed, right? All right, here the p-value is pretty high because the p-value is close to 1, which means your data, given your data means given the data you observed, your null hypothesis is very likely to be true. So what is the null hypothesis here? The null hypothesis in this problem, in this situation is the 4 observed phenotypes are following the ratio, the magic ratio of 9 to 3 to 3 to 1, right? Or you can go deeper. So why? So what is really we are testing about now? So we are really testing whether these two genes are located on two different chromosomes, whether these two genes are independently assorted during the meiosis, right? So this is what we are actually testing using this data. All right, because
Professor,
yeah.
I'm so sorry.
No.
I have a quick question. How do you know to look between 0.9 and 0.975?
Okay, good question. So let me quickly, oops. All right, so we need to go back to this slide. So first, we understand this is the statistic we calculated for this problem, right? So this is the thing we calculated for this test, right? All right, 0.46. Okay, remember this number, please. Yeah, 0.46. So that's the statistic you calculated for your data. Okay, now if you look at this line, so these numbers, so when you look at these numbers, you're going to see these numbers are monotonically increasing from here to there, right? Increasing. So this number is monotonically increasing. Do you agree? All right. So this is corresponding to the p-value. So if you look at the p-value on the top of the table, so this is monotonically decreasing from here, increasing in this way. Is that correct? All right. So now remember the numbers you calculated for your data is 0.46 or something. And this number, so if you look at this axis, so this number should be sitting somewhere here between these two numbers. Do you agree?
Yes.
Okay. That means, so the p-value, so okay, so if you got this number, the p-value should be equals to this, right? 0.975. If you got this number, your p-value should be equal to 0.9. Then you got this number. So what is the p-value? Your p-value should be something in between these two numbers, right? 0.9?
Okay.
Yeah. Does that make sense?
Yes.
Yeah.
Thank you so much.
Oh, yeah, that's my job. Don't worry. My job is to make sure everybody understands this and do well in your exams and quiz. All right. But sorry, sometimes like, because this is my first time to teach online, so I try to get myself used to this. Last time, I think like if I answer too many questions during the lecture, we will be behind of the schedule. So so if you have your question, so write down your question on your notes. So I'm gonna try to finish the slides before 10 minutes before ending time, and then I'm gonna, so during that time, so I'm gonna try to answer all of your questions. Is that okay? All right. All right. Thank you. Okay. So since the p-value is large, 0.05. So yeah, this is 0.05 is a commonly used criteria. Also in statistics, we call this a rule of thumb. So we're going to use this criteria in our genetics class. So in any tests during the exams, if I ask you to do some calculation, you compare your p-value with this 0.05. If the p-value, your p-value is greater than 0.05, then yeah, you do not reject the non-hypothesis because your data is quite supportive of your hypothesis, your model, right? So in this case, since the p-value is so large, so we think, yeah, the model is true, or we think the hypothesis, the non-hypothesis, is also true, which means the data is following the 9:3 to 3:1 ratio, or you can say, all right, these two genes are independently sorted during the meiosis, right? I hope this is clear. Okay. Okay, so this is 0.05. We're going to use this. And the summary, I think, yeah, this is the second law. Let me relaunch this. Oops. Sorry about this. Okay. All right. So the second law is talking about the relationship between two genes, right, on two different chromosomes, and they are independently assorted during the meiosis, right? Okay. So a summary for the dihybrid cross, the two genes. So di means two. So we're talking about two genes. Remember when we talk about one genes, the monohybrid. So we use 2x2 table, Punnett square, to understand the genetics. Now we have two genes, then this becomes 4x4 table like this. Right? Okay, so it's easy to understand that because why is that? Because when we talk about one genes, the monohybrid, we see the phenotypic ratio of 3 to 1, right? So this is for one gene, gene 1, and this is for another gene, gene 2. If they are independent, so we can simply calculate the cross product of these two ratios and end up with this magic ratio. So this is the same for three genes. If we think about, so we can extend this to three genes, the trihybrid. So if three genes are located on three different chromosomes, so we can multiply another 3:1 ratio here. So then you can get the new phenotypic ratio for this trihybrid cross, right? Okay, test cross. This is very important. Again, this is very important in the midterm, or maybe this the next quiz or the one after next. I'm gonna give you some questions about test cross. So what is the test cross? The test cross is a genetic tool that we can leverage to find the genotype of any individual. So let me give you an example. So this is a very simple model to do this. So if I give you a purple flower here, and if we know the color of the flower is determined by just one gene, there are two alleles, the capital P and the lowercase p, and the dominance is like this. So the big P will give you the color, the purple color, but if you have the a lowercase pp, that's the homozygous recessive, then there's no color for the flower, the white. All right, but if I give you a flower with the color, so you do not know whether the genotype for this flower is either the capital P, or the capital P and the lowercase p, right? Because either genotype will give you the phenotype. So how do we know which genotype this individual carries? Then we can use this test cross. So what we do is so we call this individual with unknown genotype as the testee. Testee, and this is the tester. Tester, ER. Okay. So why we use the little p as a tester? We call this homozygous. Oh, okay, actually, yes, here, tester, testee. All right. So why do we pour this homozygous recessive white flower as a tester? Because this individual carries the double recessive alleles. So if we cross this tester with the testee, it will not hide. It will not hide anything about the testee. You will agree? It will. It will not. So let me tell you why is this. All right. So for this tester, it can only produce one type of allele. So only the small p, right? However, so let's look at here. If this one, for this one, the genotype is big P, so it can only produce one type of alleles as well, big P. So what happened here? So the progeny from this cross, will be all purple flowers. I hope everyone agree with this. However, if this one is not a homozygous, is a heterozygous for which the genotype is big P and small p, then this individual will produce two types of alleles. Half of the alleles will be big P, and the other half of the alleles will progeny will carry the other half of the gametes will carry this allele, the small p. So then look at the progeny from this cross. Half of the progeny will be colored flower, the purple, and half of the progeny will show no color.
Professor,
Does that make sense? Yes.
I have a question. So the testy, it's called testy because all the products will be dominant, looks like the dominant.
Could you say that again? Can I? So your voice is very low. Sorry.
So the testy, it's called testy because all the product looks like the dominant.
Yes, that's right. So that we call this testy because we only see the phenotype of this one, but we do not know the genotype. That's why we want to use the test cross to find out the genotype of this one, right? So if this flower, the testy, is a purple flower, so what we can conclude is this individual has at least one capital P, right?
Yeah.
But for the other one, we do not know. So how do we know? So we can use this test cross to figure it out. There are two possible ways to do. There are two possible outcomes. If all the progeny from this test cross, showing purple flowers, then we can conclude this testy will carry the genotype of capital P and a capital P. Is that right?
Yeah, makes sense.
Yeah, if the progeny look like this, so half of the progeny are purple and the other half do not show any color, so then we immediately can conclude this testy is carrying a genotype, heterozygous genotype, big P and a small p. All right, so this is very important. Please understand this. And in the test, I'm going to give you something like this. You will help me to diagnose the genetics, the genotypes for any unknown individuals. All right, okay. So this is a simple model. We use one gene, two alleles, and we can do the test cross for two genes as well. So in this case, so suppose I gave you a round shape, yellow seeds, and so based on this, you can conclude, okay, at least this individual, this pea plant will carry at least one big Y and one big R, right? But for the other one, you do not know. So how do we definitively identify the genotypes, the genetic makeup for this pea plant? So we do the test cross using this, the double homozygous recessive. So this one is little y, little r. So, right? Okay. Now the interesting thing is, so because we do not know the genotype, the genetic makeup for this one, so there are four possible ways that this individual, there are four possible genetic makeup genotypes for this individual. You agree? All right. So let's see. So for each possible genotypes, what will be the phenotypic ratios for the progeny coming from this test cross? All right, if we look at the first one, so test cross, for test cross, we always use this, the double homozygous recessive, because the tester carrying this double homozygous recessive will not hide anything from the one to be tested. All right. So if the genotype is this, so what you're going to see is like all the progeny from this cross will show the same phenotype, or yellow round. Do you agree? There's only one phenotype. However, if the testee has the genotype like this, so you're going to see this, only two phenotypes. So I don't have time to go too deep. So I just show you this. Yeah, probably it takes a little bit of time to digest this, but believe me, this is true. So after the class, please spend some time to study this table to make sure you understand what is going on behind 4 possible test crosses. And then if you understand this, then no problem with the quiz and the midterm, right? Okay, so test cross. Okay, so suppose when you do the test cross, this is the data you got. So roughly you believe, okay, there are, so based on this data, you can conclude, okay, there are 4 different phenotypes in the progeny coming from this cross, right? Okay, and the data looks like the ratio looks like this data is following a ratio of 1 to 1 to 1 to 1, and now you can conclude. So you can hypothesize, all right? The one being tested is carrying the genotype like this, and only this test cross will gives you a 1 to 1 to 1 to 1 ratio. And okay, why is that? Because you can use this to simplify the Punnett square, not square, Punnett rectangle, to help you to understand why this is true. And then once you have the data, so you can test your data, right? Because in this case, the expected fractions for each category will be equal to 1/4, the quarter for each type, each phenotype. And then, so you have two competing hypotheses. So you're gonna put the number on a trial, use your data to test if this is following a 1 to 1 to 1 to 1 ratio. And so for example, if I give you a question like this in the midterm, it's better. So it's better to use a table like this, right? Because this is your model. Yeah, if the genotypes is like that, and if these 2 genes are independently assorted during the meiosis, you would expect to see the phenotypic ratio is following this. Okay, so this is your model. And if this model is true, so then you can calculate the expected value for each category, right? So first, you need to calculate the total, and then you multiply the total by each fraction here to end up with a value here. So this is the expected value for each category. And then you use this formula to calculate these values here, here, and here, and add up to this. Now you have your test statistics, and you have your degree of freedom, which is also equal to 3. Yeah, I hope everyone understand why degree of freedom equals to 3 in this case. And then you just use this table to find the value and the p-value to make the conclusion for your test. Then bingo, you're done with your test. Isn't that very easy? It is very easy. I mean, genetics is fun. So if you understand what is going on behind, you're going to enjoy it. Right. Okay, so I think I'm gonna move on to the third lecture. So let me give you like maybe 2 minutes break. So if you have any questions, quick questions, I can help answer here now before I start lecture 3.
I'm sorry, Professor. In slide 24.
24, yes.
You said the 110 multiplied by 1, it's gonna equal to 28.
Oh, sorry. Because we cannot use this is the ratio. We need to use the expected fraction. So which means like this, the sum of this 1 equals to 4, right? So this translates to here, the expected fraction should be 1/4.
Oh, makes sense. Okay.
Yeah, and then you multiply this, you got this. Okay. That's why you see the expected value for all these 4 categories are identical, right? Because they should be the same.
Yeah, that makes sense.
Okay, sure. Okay, so let's move on. Let me stop sharing this one. And okay. Okay, lecture 3. All right, so far in the we have covered a very important concept. So I hope everyone crystal clear about this concept, alleles, because this is very important. So when we talk about alleles, we are talking about a one gene, right? One gene. So in one genomic locus, so we have one gene, but there are more than one forms of that gene, different copies, different kinds of copies. And now we say different kinds of copies or versions of that gene are different alleles. All right. For example, this is a very simple model. So here we have one gene, but we have two alleles. This is the simple model. But in reality, for one gene, let's say this is one gene, the wild type one gene. It can so the functional wild type allele will produce some functional protein or enzyme that can do something in the cell, in each cell. However, there might be more than one mutant alleles. For example, if some mutations occurs here, so this will produce one type of alleles, right? Mutant allele. If there's another mutation occurring here, this will produce another additional, the third type of the mutant allele, and so on. So in reality, so the allele model can get very complicated. So why is that? Let me give you a very typical example here. So this is a so can you see the full screen of my screen? Because I cannot see the full screen. So all right. Okay, so this is a disease, human disease called the PKU, right? So this is one gene disorder. So the normal gene can produce the functional PAH enzyme, but the mutant, if the gene has some mutation, then that mutation can cause problem with this enzyme. And then if that is true, so the individual will have some disordered phenotype. All right, so what is this gene? So what is this enzyme doing in cell? So actually, so we eat food, and so in our food there's proteins, and the proteins can be broken down into lots of amino acids, including this one. So sometimes I have a problem in pronouncing this phenylalanine. Yeah, phenylalanine. Okay. So this is a protein, and in a normal individual, so if this gene is normal, this normal individual will produce functional enzyme, and this enzyme can break down this amino acid, then everything is okay. However, if something wrong occurs with this gene, and if an individual no longer produce the functional enzyme, then this pathway is going to be blocked. This pathway will be blocked. And what happened here? So since the phenylalanine, okay, if this cannot be break down and it will accumulate in the cell or in the body, then this will cause problem. Okay, so one gene can cause more than one diseased phenotypes. So this is a very useful concept here. So I hope everyone can pay attention. So I may give you a question about in the exam and ask you, what does this term mean? All right, pleiotropic. So pleiotropic means one gene problem, but it can cause more than one phenotypes. So for example, if this gene if something goes wrong within this gene, it will cause mental retardation. It can also cause the reduced hair or some abnormal skin pigmentation. Okay, let's look at the molecular level basis for the mutation.
Excuse me, Professor.
Yes. Yeah.
Did you say one gene problem but changes a bunch of phenotypes or genotypes?
Of phenotypes. So this, for example, if this gene has something wrong, right? If there's something wrong with this gene, it can cause more than one phenotypes. So those are the disease phenotypes.
Okay,
Different
thank you so
disorder.
much.
Sure. Yeah, no problem. Okay, so look at the wild type allele. So this is a intact gene. So everyone, I believe everyone has already taken the bio 5 series classes. So you understand the structure of a gene, right? So this is an intact gene allele, the wild type allele. It has a functional promoter. This is important because without this promoter, so the gene cannot be transcribed. And within the gene, we have like exon and the intron. So, and okay, so once the gene is transcribed, so the intron will be spliced out and all the exon will be spliced together to produce the functional messenger RNA, and then the protein can be produced based on the messenger RNA. Okay. Okay, now in this diagram, so as you can see, so some areas, so look at the exon, some areas are labeled with red, which means those regions are very important. So nothing can be changed in this region because those are very critical regions. Those regions represent the most active sites in this protein, in this enzyme. As we know, so how does an enzyme function itself? So the function of an enzyme depends on its shape. So the shape, those pink area actually are most critical region that can determine the shape of the enzyme. So if the shape is right, it can function right, and it can do the right thing in the cell. However, if anything changed in this pink region, it will change the shape of the enzyme. And if that happens, this enzyme cannot do the right thing in the cell. All right. Okay, let's look at the first. Oh, where's my mouse here? Okay, the first mutant. So this one, a mutation occurs in the promoter region. So this is a very important region. So promoter cannot be touched. So if something happened here, so the gene cannot be transcribed. That's why we call this non-mutation, because this mutation will cause a completely loss of the function of this gene. So the function of this allele will be annulled. Right. Okay, let's look at the second mutant. So this is another allele, right? We have different versions of alleles here. We have more than 2 alleles, wild type, and a bunch of mutant alleles. Not like the simple cases, the simple case we mentioned before. Here, this is more reasonable in the reality. All right, the second one, if the mutation occurs in the pink area, so this is fatal, because the mutation will occur the critical parts and will change the shape of the enzyme, and then the function of this gene will be totally destroyed. The same thing here. So we call this, although the mutant 2 and mutant 3 are different alleles, but they have the same phenotype. You do not see the functional protein. However, this one is a little bit different because the mutation occurs between the pink and the green one, which means like only, so this is not fatal, but this mutation will cause some partial loss of the function. Then you're going to see partial phenotypes of the disease and the partial normal. So we call this kind of mutation as the leaky mutation, right? Because yeah, the mutation occurs at some not that critical region. So for this one, this mutation totally occur in a irrelevant region. So this will not affect the function of this gene at all. So we will say this this mutation is a silent mutation because you do not see any consequence from this mutation. And this one is also fatal. Why? Because even this occurs in the close to the green region, but it occurs somewhere between the intron and the exon. So you know, when the gene is transcribed to form a messenger RNA, so the intron will be spliced out But if something goes wrong in that region, so this will affect the accuracy of the RNA splicing. So this can also affect the gene expression. So this is a molecular basis for how different alleles can come from a mutation, different versions of the mutation for a wild type allele. So I hope this is clear to everyone. Okay. All right, let me give you a very brief review. So first the Mendel, second the Mendel 1 and the Mendel 2. So the Mendel 1 again, so we talk about two alleles, two members of one gene during the meiosis, right? But in Mendel 2, we are talking about the relationship between two genes, and these two genes are located on two different chromosomes. So during the meiosis, these two genes are independently sorted. Okay. All right. So this is a model, the diploid model we can use to understand the dihybrid cross. So as we have learned already, so if we do the dihybrid cross, so each F1 dihybrid parent will produce 4 possible gametes here because of the independent assortment, right? And so these 4 types of gametes will have equal chance based on the independent assortment hypothesis, the second Mendelian law. And so this is the model we can use to understand the genetics behind the diploid model. And also we can use the haploid model as well. So this model is a beautiful model. So the organism is called the Neurospora. Oops. And so I don't think I have time to go over the life cycle of Neurospora, but I highly recommend each of you to read the textbook and make sure you understand the life cycle of this magic model organism. So it's a haploid model organism, Neurospora. So why this model is so beautiful? Because you cannot see here. So it can produce so-called the linear meiosis system. So because this is a haploid, only one end, right? So let's look at if a female parent is mating with a male parent, so they are gonna form a transient 2N, which is called the male site. And this male site can go through something which is very similar to the diploid organism, the meiosis. So this can produce something like the gametes, but here, the individual spores here. And it can go through an additional post-meiotic mitotic division to produce 8 individuals here. But the unique thing here is why we use this as a ideal genetic model to study genetics, because so for those resultant individuals spore here, so they cannot move around, they cannot move freely. They are actually held within a linear sac. So this is the beauty. They cannot move freely, not like the animals. So if a parent produce lots of sperm or eggs, they can move around. So that's why you cannot trace the genetics using that model, right? But here, because for any single meiotic events, the products, the resultant individuals will be held together within this beautiful sac in a linear fashion. And then so what's the benefit for this? So look at here. So if we made this parent with this parent haploid, right? So the resultant individuals will look like this. So if there's no recombination occurring between these two parents, so we're going to talk about the recombination. now. So that's for the next lecture. But here we understand, we should understand if there's no recombination occurring. So this 8 product from this mate mating will be nicely held together in this fashion. So this is why this is a very, very useful model, genetic model for us to study genetics. And in the future, probably after the first midterm, so we're going to use this model again to do the genetic mapping. So yeah, that's a very fun part. All right. Okay. So as you can see, if there's a crossing over occurring between these two parents, so the pattern will be changed, right? So in the previous slides, so you're going to see if there's no crossing over or recombination occurring between these two parents, you're going to see the big A, big A, big A, big A, small a, small a, small a, small a. They are arranged like this in this linear meiotic sac. However, if there's a crossing over occurring between these two parents, you're going to see the pattern changed. All right, big A, small a, small a, big A, small a, small a. So, okay, so this is not the topic for this lecture, but in the future, we're going to use this as a model to help us do the genetic mapping. All right, recombination. Now we need to learn a very important concept here: recombination. So what does recombination mean? Recombination means, so if we cross two parents here, the parents may have its genotype, right? And we get F1, which is big P and small p. And then we do the selfing. We will generate a lot of different progeny here. And some progeny will have genotypes very different from your parents, from these parents. And if we see some new genotypes occurring from the progeny, then we say there's a recombination. So the definition means like, so when compared to the genotype of the parents, new allele combinations occurs in the progeny. All right. So there are multiple ways to produce the gene recombination or the allele recombination. The major thing we have covered is the independent assortment, Mendel-2, right? Because of the Mendel-2, so we see brand new allele combination arise from the progeny. All right, let's look at this. There are some other mechanisms that can also cause the DNA recombination, but this will be our future topics, not here. Okay. So let me use a diploid organism as a model organism to show you what the recombination means in this test cross. Right. So here we start from two parents here. Now, the two parents only have two types of allele combination. So for the first parent, the allele combination is the capital A and the capital B. And for the second parent, there's only one allele combination, which is the lowercase a and the lowercase b. So we cross these two parents to produce the F1 progeny. The same, still, even the genotype look like this, but still we have only two types of allele combination, right? The big A, big B, small b. However, when we do the test cross, this double heterozygous individual, dihybrid individual will produce four types of gametes because of what? Because of the Mendel-2, because of the independent assortment, right? So we have learned this. So I hope everyone understand this. So because of the independent assortment, so this F1 dihybrid parent will be able to produce 4 types of allele combinations. Of course, these 2 are identical with the initial parents, right? But we see 2 new allele combinations from this dihybrid cross, right? And if we do the test cross, remember, test cross, we use an individual carrying the double homozygous recessive alleles to test across this one, and we're going to produce a progeny like this. So out of this progeny, let's see what happened. Okay, half of the progeny will show the exact phenotypes as the initial parents, right? But the other half of the progeny will show new phenotypes, because of the new allele combinations arised from this test cross. Am I clear? And because of the independent assortment, and because of the, so we talk about the 4x4 Punnett square, right? So because of this, we expect to see 50% of the progeny from this study will show the new allele combination or the new phenotypes. Right? So this is the diploid model, and in the haploid model, the same. So we can cross these 2 parents with 2 different allele combinations. We call this input. So this is the input genotype for the initial parents, and we do the we cross these 2 to form a meiotic male site, and this male site will go through the meiosis to produce the progeny, haploid progeny. And if you look at the phenotypes for all the progeny, you're going to see half of them have the same phenotypes of the initial parents. The output, half of the 50%, 50% of the output from this cross have the identical phenotypes with the input initial parental phenotypes. But the other 50% of this output will be different. Will carry a completely different phenotypes when compared to the initial input parental phenotypes. All right, this is why. This is because of the Mendel 2, because these 2 genes are located on 2 different chromosomes, and during the meiosis, 2 genes are independently assorted. Right. Okay, so test cross. So I don't think we need to go over this again. And okay, so very important concepts. So here, so far, we only cover this part because we are talking about what? The 2 genes on 2 different chromosomes. So during the meiosis, the pairing could be different. So there's 1 way, 2 way, another way for the pairing of the homologous chromosome, and this will end up with different combination of those alleles, right? So because these 2 genes are on 2 different chromosomes, so they are independently assorted. That's why we always see the 50% of the recombination progeny. So which means, so if we do this kind of test cross, we always see 50% of the output, the progeny will carry the recombinant alleles. All right. So this is what we have learned. But there's another way to produce recombinations. So compare this one with this one. Here we are talking about 2 genes on 2 different chromosomes. However, in the future, if we talk about 2 genes or 3 genes on the same chromosome, for example, the same chromosome, we have this locus and this locus and this locus, which represent 3 different genes. They are not independent at all. They are not independent anymore, I'm sorry. Why? Because they are linked together. They are on the same chromosome. So this situation is different from this situation, right? And still, like during the meiosis, there's a DNA recombination or there's a gene recombination because of crossing over. So I believe most of you have already learned or heard about these concepts. So in the future, we're going to use this. We're going to use this because see, if there's a crossing over between these two homologous chromosomes, there's a DNA swap between these two chromosomes, homologous chromosomes. So the big C moves here, and then the small c moves here. So this will also generate different DNA recombinations. Okay, now, so this is our future topic. So we're going to use this to do the genetic mapping, and we can use test cross again to help us to determine the exact location, genetic location of the 3 genes on the chromosome based on the recombination frequency. All right, so I just introduced this concept to you. This is not what we need to understand, fully understand for this chapter, but in the future, we're going to use this concept to help us to do the genetic mapping. All right. Another thing I would like to mention is, okay, so here, if 2 genes are located on 2 different chromosomes, the recombination frequency is always equals to 50%, right? This is what we have learned. They are independent. However, if the 2 genes, let's say AB, they are not on different chromosomes, they are on the same chromosomes, then the chance you're going to see the recombination between A and B is always less than 50%. It's less than 50%. Okay, so this is a criteria in the future. We're going to use this criteria to tell us whether these 2 genes are located on 2 different chromosomes or not. Okay, so some review for the probability statistical theorem. So I don't think I have time to do this. So please read those slides after class, and maybe in the discussion session, Lei can help you to go over this statistical probability theorems here to help with your future calculation. And all right, so this is the extension. Okay, so we use the 1 gene model, 2 gene model to study the monohybrid cross and the dihybrid cross. Now we can easily expand our theorem to 3 genes, trihybrid model, right? The hybrid model. So here we talk about like 3 genes. So here, the 3 genes are located on 3 different chromosomes, right? So if we cross these 2 parents, we got the F1 trihybrid, and then if we do the selfing for this one, we're going to generate. So each parent will generate this many possible gametes. So why is that? So why 8? So if we talk about 1 gene, we're going to end up with 2 possible gametes, right? If we are talking about 2 genes, then we will have 4 types of gametes, which is equal to 2 to the power of 2. And now if we're talking about 3 genes on 3 independent chromosomes, then we're going to end up with 8 possible gametes, which is 2 to the 3 power of 3. All right, that's all about the magic number of 2 because we're dealing with a diploid organism. All right, so if in the test, if I ask you to calculate the expected frequency of the progeny that are carrying the genotype like this, so how do we do that? It's easy because the 3 genes are independently assorted during the meiosis, right? So we can just calculate each genes one by one. So for gene 1, so this is for gene A. So this is something like this. the monohybrid, we use the simplest Punnett square to help us calculate the probability that the progeny will carry this genotype. So it's easy, right? So that's 1/4. So the same for little b, still 1/4. But for the heterozygous capital C and the lowercase c, so there are two possibilities. One is the progeny is has one capital C from mom, but small c from dad, or the other way around, right? So get the small c from mom and the capital C from dad. So then we have to add up this two together. So the final answer should be this. So here we're going to use the rule of addition and the rule of product. So tomorrow, I don't have time to go over those probability theorems with you, but tomorrow during the discussion, so Lei will help you to go over this calculation and those statistical probability theorems. Yeah, tomorrow. And tomorrow will be our first quiz. Yeah, so please get well prepared for that. And yeah, please use the UJA system. Okay, so as we have learned, so for one gene we have two phenotypes and three possible. So monohybrid cross, we have three possible genotypes, and the phenotypic ratio is 3:1. If we have two genes, then it becomes a little bit complicated. Three genes, and so on. Just think about if genes becomes more and more. So in human, we have like 23 pairs of chromosomes, right? If we consider one gene on each of the chromosomes, so it is very complicated. So n equals to, let's say 23. So there are how many possible phenotypes? There are this many phenotypes, and there will be this many genotypes, and then the ratio will be super complicated. So this explains why even kids from the same family, they cannot look, they could look very different from each other because of this, the independent assortment. Right? Okay, that's the recombination. So let's move on. Okay, now we just used the plant model or the Neurospora haploid model to help us to understand the genetics. So the long run for the study is to apply those genetic rules in human beings, on ourselves. So in this chapter we're gonna learn something about the autosomal recessive and dominant disease. But I don't think I have time, so like 10 minutes. Oh, 3 more minutes. Okay. So this chapter we're gonna only focus on the disease phenotypes that are controlled by the genes on the autosome, not on the sex chromosome. So you're gonna see these examples here. Oops. Okay. So when we study humans, we cannot treat humans as the pea plants, right? There's a lot of experiment or things we cannot do with humans because of the ethical reasons. So what we can, so that means like when we study humans, we have to use completely different methods. So when Mendel did experiment on the pea plants, he can do whatever what he wanted, because you can control the experiment. But for human, so we have to choose to use totally different methods. So since we cannot control any experiment in human, so what we can do is just look at family trees. We can get information from the pedigrees and use that information to help us to trace the diseased alleles in the family and in the population. Okay, so as I mentioned in this chapter, we're going to focus on the autosomal inheritance So this is irrelevant to the sex chromosome. And the pedigree means the record of a family. So for example, we're going to use those commonly used notations to help us to generate a pedigree. So for example, those are very important. So I hope everyone understand this, because we use the square to represent the male and the circle represent the female, and the mating between them using a horizontal line like this. And if solid shape means the affected individual, if you see this, which means it is a carrier, heterozygous individuals. So let me quickly, I think I'd better finish this because this is important for tomorrow's quiz. Okay, so if you look at this pedigree, so you only see like the affected individual show up in some generation, not all the generation, which means this is a recessive disease. If it is a dominant disease, you're gonna see affected individual in every single generation. So by doing this, you can see, you can determine the nature of the disease, right? Because the recessive disease can skip some generations. All right. We have learned this disease, and okay, suppose there's a couple, they wants to have a kid, but they consult with a genetic counselor because the man has a sister with the disease and the woman has a brother with the disease, but both of them are normal. There are not other known cases in their families. So in this case, we want to determine the probability that their first child will have the disease. So it will be useful to use some pedigrees to help us to do the calculation. So for example, this is the father, we use the square here. This is the mother, we use a circle because they are unaffected. They are not affected. That's why we use we do not use the solid shape, right? Okay. However, based on the information we knew, like this man has a sister who is affected by the disease. That we immediately knew the genotype for his sister. The same for the woman. So her brother also is an affected individual. So based on this information, we knew the grandparents should be carrier, heterozygous for both family. Okay. Let me remove this. Now, so if we look at the mating for this family, so this is exactly like the classical one gene, two allele, monohybrid cross, right? So for one parent, there are two possible gametes. For another parent, the same. And so this is the genotypes: big P, small p, big P, small p, and small p, small p. Okay. All right. So now we want to calculate how likely, because if this man and this woman, if they have a kid with the disease, which means both of them should be the heterozygous, just like the grandparents, right? So we want to calculate the probability that this man is a carrier and this woman is also a carrier. The trick here is like, because this man and this woman are not affected by the disease. So from this monocross, we can eliminate this possibility, right? That they cannot be the homozygous recessive. So they could be one of the three situations. Then the probability that this man is a heterozygous is equal to what? Is equal to 2 out of 3 for the man. And for the woman is also the chance that this woman is a heterozygous carrier is also 2 out of 3. So the probability of that is this. And if given they both are heterozygous carrier, what is the chance that their first child is a affected individual? So this we will go back to this Punnett square again, right? That's the chance of that is this. So if you multiply all these numbers together, that will gives you final answer. All right, so I think I'm gonna stop here. So we have 6 minutes that I can answer any of your questions here. Yeah, we're almost done, so we are in a good shape. Any questions?
Professor, where did the 1/4 come from?
Okay, sure. Let me redraw this. Okay, so this is the Punnett square, right? And this is the genotype, big P, and this is big P, small p, and small p, small p, and small p. Okay, so we want to calculate what's the chance this man and this woman will have the first child with the disease, which is the small p and small p, right? So 1/4 is here, so represent. So if you look at the cross between this man and the woman, so it's a classical monohybrid cross. So the chance that the child has the phenotype like this, small p and small p, will be 1 out of 4.
Oh, I got it. Thank you.
Yeah. Okay. So, but for each of the parents, so it's different because they are unaffected. So we will look at the same Punnett square, but we will eliminate this possibility, right? Because this parent and this parent cannot have the little p. So they can only take 1 out of these 3 possible genotypes. But here we want to calculate what's the chance that either the father or the mother will carry the heterozygous genotype. That is 2 out of 3. Does that make sense?
Yes.
Great. Any other questions here?
Yes, going back to the previous lecture regarding test crossing, and when you have the 2 flowers, if the dominant gene, which I believe is capital P, if that's present, then that's what identifies that the flower is going to be colored.
That is true, because yeah, the presence of the big P, the capital P, will determine the phenotypes, the color of the flower, because the relationship between the capital P and the lowercase p is the dominant and the recessive. So and even then, the presence of one big P will give you the same phenotype like the one carrying the 2 copies of the big P.
Okay, and then how is the P and little p determined?
Okay, let me.
Is that in a regular world? Is that through testing, or is that something that we need to know?
Yeah, good question. So that's a good question. That's so let me show you. That's a good question. So this is about the biochemistry or the molecular level of this. So for example, in this case, let me see. So the big P actually represented the wild type allele of the gene, and this gene can produce the purple pigment. So that's why if you have a big P, which means this gene is functional and it can produce the purple pigment, and this pigment will be deposited in the flower, then you're going to see the purple, right? However, if there's a mutation occurring here and the gene function has been destroyed, so the little p is here, another allele. So this cannot produce any functional, they cannot produce the pigment, the purple pigment, then then this is the difference between this two allele. So if an individual has like 2 big P, the wild type, there's no problem. It can produce the pigment for the flower, right? If an individual has a big P, but a small P here, it is still okay, because even this allele is non-functional, but this one can produce enough pigment to show the color for the flower. So that's still good. So that's the.
So then okay, so I understand that. Looking at the previous slide, the one that comes before this one, when
This one?
when we have, yeah, because I was trying to set up the table for this alone, and I was having a difficult time. Because this is similar to the one we just went over, right? We're trying to determine dominant and recessive genes.
That's right. So actually, this one is basically similar to this one, but it's a bit more complicated because we are dealing with 2 genes, right? In this one, we are dealing with just 1 gene, so 2 alleles. That's simple.
Right.
But for here, we have to deal with 2 genes. For each of these 2 genes, we have 2 alleles. So that's why we have like a little bit complicated, more complicated table here.
For the purpose of testing, I'm guessing we're gonna have to eventually be able to do something like this.
Yes.
But for now,
unfortunately, yes.
will we? Okay. But for now, are we just going to be tested on 1 feature as opposed to 2 like this one?
Right. So probably for actually so the reason why I use this simple model, because it's easy to start with this one, right? But eventually we're gonna deal with the situation like this. And I believe in the quiz and in the midterm, so you're gonna see problems like this, not just 1 gene. That's too simple.
Okay.
Yeah.
Understood. Now, if we were to go to 2 slides over, and then we're looking at
2 slides over.
the
mean
one
here?
that says, "What if yellow?" No, oh, sorry, I meant 2 slides over to the right, or 2 slides over forward.
Oh, forward. Okay,
one past the test. Yeah, sorry.
Oh, this one.
Yeah, the one. Yeah. So if we were to try and set up the table for this one, would we have on the first column? Would it be YY, RR, and then I guess I'm just trying to understand how to set up a table for this.
I see. So I think I think you are asking the format of the question shown on the exam, right? Okay. So I'm going to give you something like this actually. So
Okay.
So because this one is very clear, because this is the double homozygous recessive. So you can immediately tell the genotype for this one. But for this one, it's a little bit complicated because this one you do not know. You only knew like this one has at least 1 capital Y and 1 capital R, but for the other one you do not. But in the exam, I'm gonna give you the outcome of the experiment. So if you see the cross, the progeny from this cross gives you this 4 types of phenotypes, then immediately you knew the genetic makeup for the green one, right? So only this genetic makeup can give you 4 different phenotypes. Is that correct?
Yeah, and just to verify, so that that bottom that dash that just signifies unknown.
Yes.
Okay.
So I'm not gonna tell you about the genetic makeup for this. This is what I will ask you. You need to figure out based on the data I gave you. So we just do the cross between this one unknown, between this one with unknown genotype, with this tester, and this is what we got from this cross. And based on this data, you need to figure out the genetic makeup for this unknown, just like this. So there are 4 possible ways, right, to have a phenotype like this. But which one is the real genetic makeup for this individual testee? So you have to determine the genotypes for this one using the resultant phenotypes here.
Okay.
Does that make sense?
Yes, it does. Thank you.
Okay, sure.
I
All
was,
right.
I
Yeah.
was just wondering, would you be able to upload this like today after class just to review it before the quiz if we want to see anything?
I'm sorry, could you say that again?
Oh, would you be able to upload this video like right after class just so we could review it before the quiz?
Yes, sure.
Okay,
So
thank you.
probably it takes like about like 1 or 2 hours. I'm gonna
Okay.
do this right after, because sometimes like YouTube, it takes a little while to process.
Mhm.
But yeah, I will definitely, probably like around like 2 o'clock, I will upload the video. Yeah, to
Thank you so much.
Sure, sure.
Have a good day, Professor.
You too. Thank you. Okay, thank you. Bye-bye. See you on Thursday. Good luck tomorrow, Mr. Quiz.
Professor, I had one more question.
Yes.
It's kind of regarding the format of tomorrow's quiz. Is it going to be like multiple choice, written, or like how many questions will we have?
Let me think. So not too many, because we only have like 10 to 15 minutes to do that, right? So we have like about like 3 to 4 questions, and it will be the short answers format. So I'm gonna give you a problem, and you need to like either to give a short answers to that question, or you need to do a little bit of calculation to find the right answer. So not the multiple choice. The only multiple choice format will be used for final. I think even for the midterms, 2 midterms. So I think it's better to use the short answers.
Okay, thank you.
Sure. Any other questions? All right. Thank you. Good luck tomorrow.